Good day, everyone. This is Julian Resos Leroy from the Department of Plant Breeding and Genetics. And please join me as we review this topic here, crop improvement and seed selection. So in this particular slide, I will show you the outline of the topics that we will discuss today. So number one, we will start with the concepts of crop improvement. Second, basic steps of crop improvement. Third, germplasm. Fourth, breeding and selection methods. Fifth, varietal registration. Six, types of cultivars. Seven, concept of seed selection or certification, rather. Then eight, classes of seeds. So let us first define what is crop improvement. So crop improvement refers to any form of genetic alteration in plants to satisfy human needs. And there are two approaches of crop improvement. You have the conventional approach, which refers to the methods of crop improvement, which uses proven and tested older tools in manipulating the genetic makeup of the target plant. So conventional approach is also known as the traditional or classical plant breeding. Then you have here the non-conventional approach. So this uses cutting-edge technologies for creating variability that is sometimes impossible to achieve with conventional methods. Then, under conventional approach, you have hybridization through controlled pollination. Then you have plant introduction. This refers to the process of importing new plants or cultivars of well-established plant from the area of their adaptation to another area where their potential is evaluated for sustainability for agricultural or horticultural use. Then you have here traditional phenotypic selection. This refers to the discrimination process which based on the appearance or visible trait of crops. And then on the other hand, in the non-conventional approach, so you have the mutation breeding. So this refers to crop improvement approach which make use of mutagenic agent in creating variability, such as physical mutagens, such as ionizing radiation, and chemical mutagens, or also which include sulfonates and other chemicals that induce mutation. Then you have here recombinant DNA technology, so also known as rDNA technology. This is a powerful tool for genetic analysis, manipulation, and this tool facilitates gene transfer across natural biological barriers by bypassing the sexual process in plants. Then you have here, last but not the least, the marker-assisted selection. So this make use of molecular markers to increase the efficiency and effectiveness of the selection process. Okay, so let's discuss the basic steps of crop improvement. So number one, you have to set breeding objectives or setting breeding objectives. So this involves deciding what target environment or what target trait to improve in a certain plant. Then second, assembly and maintenance of germplasm. So germplasm refers to the collection of plants or seeds having diverse alleles of all the genes of a given crop. So in improving crops, so you need to have a wide, diverse germplasm so that you have many options of the genes to be pulled in your breeding program. So next, you have creating variability. So this involves the conventional and the non-conventional tools in creating variability, and we will discuss it further later. And then fourth, you have selection. This refers to the process of discriminating among genetic variability to advance in a fraction of the next generation or the breeding cycle. So you have to have a breeder's eye to select which is which. So to select individual plant which inlines or answers your breeding objective. The next, you have number 5, evaluation. So this refers or also refers to the selection process which involves comparing a set of superior candidate genotypes to selected one to be released as a cultivar. Okay. And then last but not the least, release of cultivar. So that is the ultimate goal of the plant breeding, is to release a new cultivar. Either it can be released nationally or regionally released. Okay. Breeding objectives in crop improvement is geared towards fulfilling human needs. So here are the current issues faced by human, and here are the crop improvement objectives based on answering these current issues. So number 1, growing world population. So as the world population grows, it means that there are more feeds or more mouths to feed on. So we have to increase crop yield production as one of the crop improvement objectives. The next, low food and feed nutritional content. So to combat this issue, you have here biofortification in plants to improve the nutritional contents of food and feeds. Then you have harsh environmental conditions. As we all know, we are facing global climate change. So the environmental condition or growing condition of plants becomes unfavorable. So you have here development of biotic and abiotic resistant plant varieties, and then increased demands of new horticultural plant varieties, especially during the pandemic. So there are more plantitas and plantitos who are looking for new plant varieties which are unique from the eyes. So you have here development of new horticultural or new and unique horticultural plant varieties. Then fifth, you have different specific production systems. So these production systems needs different types of plants. So here, development of plant varieties suitable for different production systems. So especially on highly mechanized agricultural area in other advanced countries. So they need special crops or plant varieties that can survive with the rigors of the machines. Then you have here 6 different end use requirements. So here, development of plant varieties suitable for different end use requirements. Like for example, if you are producing tomato for table, for industry and for family consumption. So there are different end use requirements of different tomatoes. So if the industry are making tomato paste, so they have different requirement for that specific tomato. For tomatoes that are used as spices, so there are also different requirements for that. So in here, you have different plant varieties suitable for different end user. Okay. After discussing most of the possible breeding objectives in crop improvement, now let's proceed to the germplasm. So again, germplasm refers to the sum total of all hereditary materials in a single interbreeding species. So germplasm is considered as the lifeblood of plant breeding since it provides the material to be used to initiate in a breeding program. Okay. So this germplasm includes the following materials. So number 1, you have domesticated plants. So these includes plant materials that have been subjected to some form of human selection and are grown for food and other uses. Then under domesticated plants, you have commercial cultivars. So this includes current cultivars, retired or obsolete cultivars. Then you have here breeding materials. These are intermediate breeding products which usually has narrow genetic base because they originate from a small number of genotypes or population. Then you have landrace. These are farmer-developed and maintained cultivars which have co-adapted gene complex. Then you have plant introduction. So this includes unadapted genotypes from outside the production region, which usually from another country. Then you have genetic stocks. These consist of products of specialized genetic manipulation by researchers, such as genomic mutants. And then you have here undomesticated plants. So this includes wild or weedy relatives of domesticated plants. Then last but not the least, other species or genera. So these plant materials can be grouped into one germplasm. Okay. So after discussing germplasm, so let's proceed to genetic variability. So creating genetic variability refers to the differences between species, breeds, lines, or cultivar due to the following sources of genetic variation. So you have three. Number one, you have recombination. Number two, mutation. Number three, migrations of genes. So let us first discuss the first source of genetic variability, which is recombination. So these refer to the process which progeny derive a combination of genes different from that of their parents. And there are two approaches of recombination. So you have the conventional approach, which involves controlled hybridization or cross-pollination using different mating systems. And the second approach is the non-conventional approach, which involves gene transfer and soma clonal variation. Under conventional approach, so you have here mating designs. This refers to the artificial crossing or mating scheme in plant breeding programs. And you have here, it has two primary purposes. So number one is to generate information for the breeder to understand the genetic control or behavior of trait of interest. And second, to generate a base population to initiate a breeding program. Under this mating design, you can produce hybrid crosses. So these are produced through the following. So you have single hybrid cross or single cross. So in here you have two parents, A and B, and then you have to cross that one, producing an F1 called AB. Then you have also a three-way cross in which you have three parents. So first you have make a single cross. So A and B, then you have F1, and then the F1 of A and B will be crossed to the third parent, producing, you have here a three-way cross hybrid or ABC. Then you have also a back cross. So in back cross, you have here A and B parent crossed together to produce F1, and then one of the parent will be crossed back to the F1. So in this case, you have letter A, parent A, so is crossed back to the F1. So now you have BC1 or back cross 1. Then you have double cross. In here, you have four parents. So you have A and B cross together to form F1 AB, and then C and D cross together to form CD, F1 CD, and then the two F1s will be crossed together to produce A, B, C, D F1s. So you have here double cross. Okay. So here under mating design, so you have biparental mating or pair crosses. So this is the simplest of the mating designs. These involve selection of large number of plants that are random and cross them in pairs to produce full seed families. So it has three designs. So first you have the North Carolina design 1, North Carolina design 2, and North Carolina design 3. So in here you have the first North Carolina design or the North Carolina design 1. So in here, this design is a nested arrangement of genotypes for crossing where each male is mated to a different groups of females. And in letter B, you have here the practical layout in the field. So in here, in letter A, so here, as you can see, you have here the males, A, B, and C, and they are mated to different groups of females here. So you have the first group, you have D, E, F. The second group, you have G, H, I, and then the third group, you have J, K, L. So those are the females, different females, and here are the progenies produced from this biparental cross. And then in the field, you can lay out here like this one. So you have here the group of females here, and you have here the males. Okay. So this is the North Carolina design 2. So in here, this is a factorial design. So as you can see here in the design A, so you have here the sets of males, so A, B, C, and then each males are mated in the same group of females. So you have here this group, you have D, E, F, and this group also have D, E, F. Also here have D, E, F. So these are the progenies produced here. Okay. Then in the field, you can arrange here, you can arrange these progenies according to these lines. So you have A, D, A, E, A, F. Then you have B as the male, A, uh, B, E, and then B, F. Then you have also C, D, C, E, C, F. Okay. So this is the third or North Carolina design 3. So in this design, a random sample of F2 plants is backcrossed to the two inbred lines from which the F2 was descended. Then you have in the A, you have the conceptual form, and B, the practical layout, and letter C, you have the modification. So in here, as you can see, in this part, so you have here the A and B, these are your two inbred lines here. Then you have generate F1, and this gen F1 will be crossed by itself to produce the F2. And then here, in this conceptual approach, so you have here the A parental lines and the B parental lines crossed with the F2. And then for the practical approach or practical arrangement or layout in the field, so you can arrange in this one. So in between the A and B, you can place F2s, then also here F2s, and also here F2s. And then for the letter C, you have here the modification of this North Carolina design tree. Now we are done with the conventional approach using the controlled pollination. So you have here we have discussed the different hybrids and also the biparental matings and the different mating designs under it. Now let's proceed to the non-conventional approach in generating variability or recombinations. So number one, you have here gene transfer. So this involves recombination DNA technology or rDNA technology, a state-of-the-art gene transfer which generates genetic variability for plant breeding. So it involves transfer of genes using this state-of-the-art technology. So without the use of natural cross-pollination, so you can transfer from one gene of a specific animals or plants or organisms to a target plant. Okay, so nowadays there are many technologies facilitating this one, and all those technologies are called recombinant DNA technologies. Then you have here another source of variation, non-conventional approach. So you have the soma clonal variation. So this refers to the heritable variation among in vitro cultured plants or tissue cultured plants, which is characterized by the following changes. So you have here karyotypic changes. So change in the karyotype of the specific plant, then cryptic chromosomal arrangement, then you have somatic crossing over, then you have sister chromatid exchange, then you have transposable elements, and you have here gene amplification. So all of these takes place in vitro cultured plants. Okay, even though they are vegetatively propagated, so normally they are true to type, but under some circumstance, especially when they are undergoing different subcultures, so they can undergo these changes in their karyotype, and also changes in their genes. So you have here gene amplification. So all of those are generally called soma clonal variation. Okay, aside from uh, recombination as source of genetic variability, so you have here another source of genetic variability. You have number one, mutation. So this is the ultimate source of biological variation. And then this refers to the permanent changes in the genetic material involving either a physical alteration in the chromosome or a biochemical changes underlying DNA molecule. And you have also here another source of genetic variability, which is migration. This refers to the entry of individual into an existing population from outside. So it occurs via pollen transfer or gamete migration. And plant breeders employ this process to change the frequency when they undertake introgression of genes into their breeding population. So migration, so it also involves introducing genes into an existing population. So through, you have here pollen transfer or gamete migration. So to summarize, under creating genetic variability, so you have here number one, recombination. And the recombination, you have two approaches. So you have the conventional approach, which involves the process of hybridization. So you have here hybrid process, and you have also here the biparental matings. For the non-conventional approach, we have the gene transfer and the soma clonal variation. And then another source of genetic variability, so you have mutation and then migration of genes. So the next steps of crop improvement after creating variability is to select which variability is suitable or can answer to our objective. So you have here selection. So this refers to the process of discriminating genetic variability and advancing selected individuals to the next generation or the breeding cycle in order to develop new potential cultivars. And then it is the most fundamental techniques used by plant breeders throughout the ages. And it involves the use of standard selection or breeding methods suitable for species and breeding objectives. Okay, so let's proceed to the first selection method. So you have here mass selection. So this is the oldest method of breeding self-pollinated crops, but also it is applicable for cross-pollinated crops. So mass selection aims to improve the population by increasing the gene frequency of desirable genes in the population, and this discriminates individual based on plant phenotype. So you have here in this particular layout here, so you have uh, two mass selections. So you have first mass selection for cultivar development. So you have here the source population. So you can do two things here. If the individuals desirable plants are many, you can rogue out the undesirable plants instead, and then after roguing out or eliminating the undesirable plants, you can harvest the desirable plants. But if the desirable plants are few, so daghan ang mga undesirable ones, so ang una pwede nimong buhaton is to select the desirable plants first or harvest first the desirable plants, and then ipabili nimo ang mga undesirable one. So after selecting the desirable plants, so in the following year, you can plant those in replicated trials. So in here, and then after that one, you have to select the best performer, and you can release that as a cultivar or a variety. And here, you have here the second, so you have your mass selection for purification or cultivar purification. So you have here the source population. So it consists of 500 to 100 desirable plants here. So you have to plant that. Then after that, you have to collect 200 plants and then plant it in space with particular space here. Then you have to rogue out the off types or undesirable ones. And then after roguing out the undesirable ones, you can harvest or plant it in the third year here. And then you have to bulk harvest it. So when you say bulk harvest, you have to harvest all the seeds and mix it together. So that is bulk harvest. So here, this is mass selection for purification of cultivar. So this can be employed only if, for example, you have a cultivar and then sometimes after harvesting, nahaluan siya og lain nga variety. So kung nahaluan siya, pwede nimo siyang buhaton ini ang mass selection. So imo siyang itanom and then tan-aw nimo, patubuon nimo ang nangalahi, then ang kanturang imong cultivar, target cultivar mo ra to imong i-select, then ang katong mga lahi or off types or undesirable one i-rogue out, and then after that, after ma-rogue out, so mo na ni itanom nimo sa third year, then sure na ka nga wala na nasagol, then you have to bulk harvest it. So that is for mass selection. So you have here the second selection method. So we have here pure line selection. So the theory of pure line was developed in 1903 by a Danish botanist called Johansen. So pure line selection entails repeated cycle of solving following the initial selection of a mixture of homozygous lines, and it produces line cultivars with narrow genetic base and that tend to be uniform in trait of interest. Okay, so in here, you have here a layout or outline of pure line selection. So in the first year, you have here 1,000 seeds. And then you have to plant it. And then in the following year, you have only to select 200 plants from this one. Then plant it in progeny rows of superior plants. Then you have to compare each superior plant. Then you have to select only few plants or superior plants from superior row. So out from 200, you only select 25 to 50 for the next 3 years. So you have third, fourth, fifth year. So ang imo rang i-select ana is 25 to 50 individuals from here. And then in the 6th year, you have to plant it. So only 15 individuals na lang ang imo i-select. Then you plant it in preliminary yield trials. So in replicated trials here. And then in the 7th to 10th year, so only 10 individual plants will be included in this one, and this will be on advanced yield trial. When you say advanced yield trial, it is tested on different locations, and then there are several traits that will be measured or parameters that will be considered in selecting this. Then after that one, out from the 10, you only select 1 or 2 cultivars to be released for this pure selection. So let's proceed to the third selection method. So you have here the pedigree selection. So this is a widely used method of breeding self-pollinated crops, but not also just limited to self-pollinated crops. This can be also used in cross-pollinated species. So it employs hybridization to generate variability, and pedigree selection needs intensive record keeping of the ancestry of the cultivar. Okay. So unlike for the first two types of selection, the mass selection and the pure line selection, those two did not employ or did not involve the creating of variability. So take note, katong mass selection and pure line selection, you only start with the seeds. You are not crossing to produce the seeds. So nanay seeds daan, nanay population daan. But in here, in pedigree selection, you employ hybridization to create variability. So in here, as you can see, in this particular outline here, so you have P1 and P2 crossed together. So you have here uh, you are employing hybridization in here in this part. And then, after that one, you create F1. So, itanom nimo ang F1 in the following year. So, F1 consists of 150 to 100 seeds. And then, you have to bulk that seeds. So, in the F1 here, imo ni siyang self-pollinate ang F1. So, in self-pollinating the F1, you will produce F2s. So, many F1s nga gi-self-pollinate, you can create here as much seeds as 2,000 to 5,000 seeds. Then, you plant it in the third year, i-plant nimo siya in space, na siya kanya-kanya nga spacing. And then, in the fourth year, you have to select plants. So, 200 plants from those from 5,000. So, 200 plants, superior plants ang imong i-select. Then, after that one, you have to select down your selection to 100 superior individuals. And then, for the 6th and 7th year, so magpili na lang ka from 100, nag-close down ka into 50 or 25 to 50. And then the 8th year, only 15% will undergo for preliminary yield trials. And here, in the 9 to 11th year, 5 to 10 plants lang ang imong isulod under advanced yield trial or in more replicated trials and several locations and years. So, imo na siyang i-undergo og advanced yield trial. Then, lastly, you only release one cultivar for this under pedigree selection. So, in here, you need to have records, ancestry of the cultivar. So here, dapat makahibaw ka kung unsay parent ana niya, then asa siya nga line. So intensive nga record keeping ang kinahanglan under pedigree selection. So makakita ka kung asa na siya gikan nga lines diri, unsa nga plant dinhi siya. So makita na nimo under pedigree selection. Okay? So you have here for the fourth selection method. So you have the bulk selection. So the strategy of crop improvement which natural selection affects is solicited more directly in the early generation of the procedure by delaying the stringent artificial selection until the later generation. So in here, in bulk selection, during the first year of bulk selection, ang imong gi-allow ini is natural selection. So kung imong gi-allow ang natural selection to select which plants are suitable. Okay. And then stringent artificial selection will be done in the later years. So here, so in this particular presentation here, so you have here the cross generation of variability here, and then you have the F1 generation here, and then F2 generation. So here, so as you can see in these following years, wala kay selection nga gibuhat, artificial selection. So from year 2 to year 6, no artificial selection is employed, but only you allow natural selection na mahitabo. So for example, natural selection involves nga tahay na particular seed nga wala ning tubo. Okay. So it can be accounted for natural selection. So di na rug out siya. Okay. Kaya wa siya ning tubo or naay plant nga wala kabunga kay nasakit. So kadto siya can be attributed to natural selection. Selected out siya. Okay. And then in the years 7, 8 and so on and so forth, so you will do here the artificial or stringent selection. So kung base na kung unsay phenotype nga imong gusto or phenotype nga gusto sa breeder. So you will do selection here in the 7th year, 8th year, you will do preliminary yield trials, and in the 9th to 11th year, you will do advanced yield trial. And then after this one, in this bulk population, you will only release one cultivar for this. Okay. So that is for bulk population. Next selection methods is the single seed descent. So it was born out of a need to speed up the breeding program by rapidly inbreeding a population prior to beginning individual plant selection and evaluation, while reducing a loss of genotype during the segregating generation. So, advance the maximum of plant in F2 through the F5 generation. So here, from the term itself, single seed descent. So, usa ka seed per plant ang imong i-advance through generation. So, each individual plant will be represented by single seed sa kada generation. Okay. So, mo nang gitawag siya single seed descent. So, in here, in the layout, so you will start here generating variability by crossing P1 and P2. Then in the F1, so you have here, you have to bulk the F1s in here. And then bulk all this, bulk and space plant F1s. Then bulk and plant the commercial seedling rate. So muna, commercial seedling rate. Then here, bulk and plant commercial seedling rate gihapon. So in here, as you can see, the number of plants per year is not reduced because each individual plant will be represented by one seed. Okay, so as you can see here, tanawa, wala na reduce ang number. So here, you have also 2,000 to 3,000, 2,000 to 3,000, 2,000 to 3,000, and then here, ning dako pa siya. Nimo siyang 3,000 to 5,000. Okay. Then after that, where every plant is represented by single seed, in the 7th or 8th year, nga nakamag selection. So you have that. So select and establish the family rows from plant or heads. So you have here from 5,000 nahimo ng 500. Then here, in the preliminary yield trials, so you have 30 to 50. And then for the advanced yield trials, so you have 100. And then to be released as a variety, usa na lang. So you have your cultivar, one cultivar to be released as variety. So that is single seed descent. So now let's proceed to the 5th selection method. So you have here the recurrent selection. So this is a cyclical improvement techniques which aims to improve the performance of the population with respect to one or more trait of interest, such as a new population is superior to the original population in mean performance and in the performance of the best individual within it. So you have here 3 consists of the main phases. So first, you have individual families are created for evaluation. So parents are crossed in all possible combinations. Then you have here individual families are evaluated for new sets of parents selected. Or individual families are evaluated and a new set of parents are selected. Then you have here 3, third phase. Selected parents are intermated to produce the population for the next cycle of selection. So, so these are the main phases of the recurrent selection. So number 1, individual families are created for evaluation. And then number 2, plant or families are evaluated and new sets of parents are selected. And then number 3, selected parents are intermated to produce the population for the next cycle of selection. So you have here the 6 selection methods. So you have the family selection methods. So are characterized by 3 general steps. So first, you have the creation of family structure. Second, evaluation of families and selection of superior ones by progeny testing. And then third, you have the recombination of selected families or plants within the family and create new base population for the next cycle of selection. So under these family selection methods, you have sieve family selection, you have full sieve family selection, and you have self or S1 or S2 family selection. So now, this is the Hapsiv family selection uh, outline. So here, as you can see, in the first season here, so you have the source population. And then second season, so grow the progeny of Hapsiv here. So you have to grow the progeny of the Hapsiv. Then in the third season, you have to bulk the superior progenies in here and grow in isolation, or you have to bulk the remnant seeds of the selected superior plants. So here from these two, nagpili kag superior plants ana, pero ang imong gigamit nga seed is kadtong seeds nga gikan dire sa source of population. So remnant seeds, kauban nga mga seeds sa imong gitanom dinhi. Okay. So, in Hapsibs, di man na nimo tanan itanom. So, na kay gipabilin. So, here, pwede nimo nga ang gamiton ang seeds dinhi directly. So, mao ni ang first, mao ni ang other step. Or, you will use the remnant seeds of the selected plants from here. So, if kini nga plants imong giselect, kini siya mo imong giselect, so mangita ka sa iyang remnant or kauban nga Hapsib dinhi as sa source population. And then, mao siya imong itanom in nagka third season. And then that population here, remnant seeds of selected superior plants according to progeny test are grow in isolation, and that will become the C cycle 1 population. So from cycle 0, so you are creating now cycle 1 population. Okay. So in here, you have here the full sieve family selection. So in the full sieve family selection, so you have here the source population in here. And then you have to do crossing or select and cross pairs of parents in reciprocal process. So around 100 to 200 plants ang imong gi-cross. And then in the following season, you have to grow in replicated and test cross progeny rows. And then in the third season here, the composite of equal amounts of the remnant seeds of the superior test cross progenies grow in isolated blocks. So in here, sa ang imong maselect dinhi ang test cross progenies, so ang remnant nga seeds, magkuha kag remnant seeds gikan diri sa source nga dinhi nga population. So magkuha kag remnant seeds, then you have here equal amounts of remnant seeds. So for example, in this season 2, you selected 5 plants. So equal amount of those 5 plants seeds adto nila will be composited and planted in third season in isolated blocks. So for example, so na kay 5 ka plants nga na-select, and then ni-decide ka nga kada 5 ka plants you will get 100 grams nga seeds ana. So you have all 500 grams nga seeds from those 5 ka plants. Okay. So, composite mo na ibut pasabot sa composite equal amount of remnant seeds of superior selected test cross progeny are grown in isolated blocks. So, in the third season, it-grows siya in isolated blocks. So, meaning, isolated block. So, walay kapareha niya nga tanom diha sa iyang palibot. So, mo na siya for this full seed family. So full sieve in the sense nga you know here kung unsa ang iyang maternal and paternal nga parent. So mo nang nakahibaw ka nga full sieve na sila. So you have here cross pairs of parents. Okay. So in here you have here selection method, family selection method. So you have self family selection. So in here, in the first season, so you have the source population. So it is composed of 50 to 100 plants. And then you allow self-pollination. Then you keep the remnant seeds. And then you plant that self-seed here and grow in replicated trials. So to test progeny test here, identify superior progeny. And in the third season, composite of equal amounts of the remnant seeds of superior progenies will be grown in here and grow in an isolated random for random mating. So here from Dinia, you have self-pollinated here. So nakay source population, you allow self-pollination and then you keep remnant seeds. So naglain ka og seeds, nagpabilin kag seeds diha. And then you allow these S0 seeds or self-seeds to be grown under in season 2. Then out from that, imong gipamilian og usay superior nga progeny dinhi sa S1 progeny. And then out from the superior S1 progenies here, ning kuha ka og remnant seed anang S1 progenies, so remnant seed dinhi. Then equal amount of those remnant seeds imong gitanom in the third season. So para gahap ganina sa katong full sieve family selection, so equal amount of remnant seeds of the superior progenies and grown in isolation for random mating. Okay. So you have here number 6, family selection based on test cross. So the key feature of this selection approach is to improve both population per se and as well as its combining ability. So family selection based on test cross includes number 1, you have hap sieve selection with progeny test. So the hap sieves are tested based on the performance of their progenies. Then you have Hub-Sieve selection with test cross. So the Hub-Sieve lines are selected based on the test cross evaluation using inbreed tester. And then you have here Hub-Sieve reciprocal recurrence selection. So the Hub-Sieve are selected based on the test cross evaluation using S1 plant tester. After discussing all the selection methods, so now let's proceed to the next step, which is evaluation. This refers to the process of assessing the performance of newly developed lines through a series of appropriate trials and tests. So this consists of the following stages. So first, you have observational test. This is done by the original plant breeder and it is conducted in separate groups of experimental lines using incomplete block design or triple lattice with 2 to 3 replication in one location. So that's the first test or evaluation. And then next, you have the second evaluation, which is the preliminary yield test. This is done also by the breeder's level and conducted using check variety in an incomplete block design with 2 to 3 replication in at least 2 locations. The third evaluation procedure will be the general yield test. This is performed by a breeder or any breeder using a random complete block design or randomized complete block design with 3 replication in at least 3 locations. Fourth will be the advanced yield trial. This is conducted under the supervision of the National Cooperative Testing Level using randomized complete block design with 4 replication in 6 to 10 locations. So those are the evaluation procedure undergone by the variety before it will be released. So before a variety being released as an being called as a nationally released variety or a locally released variety, it will undergo this process, the varietal registration process. This is governed by the National Seed Industry Council, or the NSEC, through the Republic Act 7308, otherwise known as the Seed Industry Development Act, as enforced in 1992. So first step in registration process is that a genotype or a certain line must be recommended by the Technical Working Group, or TWG, as an entry for NCT, or National Cooperative Testing. So it must be recommended by the Technical Working Group. By the way, this Technical Working Group are consist of scientists or researchers that are on having the expertise of that particular variety. Okay. Next, you have here the genotype must surpass the check, or at least have the special trait as a major reason for release. So the performance of your variety, or the one that will be registered, must surpass the check. So check variety, usually these are released variety that is known to be high yielding of the moment that you are registering your crop. Okay. So it must at least surpass the check, or if it cannot surpass the check, at least your variety must have an special trait to make it a justifiable reason for the release. Okay. Next, the developer or the plant breeder of the genotype must defend and satisfy the queries of the technical working group judges about his and her crop. So in this part, the breeder will provide information to the technical working group about his crop and the performance of his crop throughout the evaluation process. Okay, for the fourth process is that the NSEC Secretariat will further evaluate the field data or coming from the different NCT sites, having all those data, and they will evaluate that and other supporting documents of the genotype before it recommending to the DA Council. So they will evaluate the data, and after evaluating the data, they will recommend that particular genotype to the DA Council. Then last but not the least, the plant breeder will present the data with the photo documentation or the evidence of the genotype to the DA Council with the NSEC personnel for the final defense of his or her crop. So in this time, the plant breeder will justify why his or her crop is ready for release or must be released as a national or as a regional variety. So after this, if the DA Council and the NSEC personnel were satisfied with his claim or the plant breeder's claim, now the variety will be given an NSEC name and also will be released either nationally or locally, will be released as a variety nationally or locally. Okay. So now let's proceed to the different types of cultivars. So first, you have here pure line or inbred. This is developed through self-pollination, and due to this process, they have narrow genetic base. Then second, you have the open pollinated variety. This is developed through natural cross-pollination, and due to this process, it is genetically heterogeneous and heterozygous in nature. Then letter C, you have here hybrid. These are produced by crossing inbred lines that have been evaluated for their ability to produce hybrids with superior vigor over or above those parents used in the cross. So you have here clonal cultivars. This is developed through hybridization and can be propagated vegetatively. So for the clonal cultivar, they are developed through hybridization, and you can reproduce them or propagate them through vegetative cuttings or vegetative propagation. Then you have here apomictic cultivars. These are produced through seeds that are genetically identical to the mother plant. So basically, the seeds here does not involve that does not involve fertilization. So basically, if the seed are not involved in fertilization, so they are genetically the same with the mother plant. Okay. Then next, you have here the multi-line cultivars are developed through self-pollination and consist of mixture of specially developed genotypes called isolines, or also known as near isogenic lines, because they differ only in one or a single gene or a defined sets of gene. Okay. Then you have here synthetic cultivars defined as advanced generation of cross-fertilized mix or mixture of seeds here, seed mixture of parents that may be strain, clone, or hybrids. Then you have here composite cultivars refers to the mixture of less similar genotypes. By the way, the last three cultivars, the multiline, synthetic, and composite cultivars, these are mixtures, but they differ on the composition of the mix. Okay, so for example, in multi-line cultivars, these are mixture of isogenic lines. So these isogenic lines, they are phenotypically the same, but they only differ in a single gene. Okay, and for the synthetic cultivars, they are mixture of cross-fertilized seeds that can be strain or clones or hybrids. So um, mixture of seeds. So this synthetic cultivars is a mixture of seeds. So um, more or less, they are also phenotypically the same. But in terms of how the same as with multilines, so multilines is very similar, Jude, ang multilines because they only differ in a single gene. But in here, it may be differ in many genes. So strain, clones, or hybrids, so they may differ. They have a little bit difference from one another. So the last, for the composite cultivars, these are mixture of less similar genotypes. So among the three, the composite cultivar is the mixture of very different cultivars or very different lines or genotypes. So because as you can see here, this involves mixture of less similar genotypes. So among the three, ang pinaka lahi-lahi o component is the composite cultivar. So after talking about the different types of cultivars that can be released in the Philippines, so now let's proceed to the process of seed certification. So before a released variety will be distributed to the farmer, so first you must have to increase the seed or reproduce the seed. So in that reproduction process, before the seed will be distributed to the farmer, it must undergo this seed certification process. So this is a legal mechanism established to ensure the pedigree of the seed produced by the plant breeder reaches to the public in its highest quality, original genetic identity, and highest genetic purity. So in the Philippines, this is governed by the Bureau of Plant Industry under their office called National Seed Quality Control Services, or the NSQS, as the seed certifying agency of the Department of Agriculture. And then BPI shall enforce rules and regulations on the seed certification through the regional office or regional field offices of the Department of Agriculture. So now let's talk about the seed certification process. By the way, this seed certification process, the applicant of this seed certification process can be any farmer that have the resources and the capacity to undergo this seed certification process. So not only the breeder. So some also they are only farmers and they are not necessarily the one who breed that particular seed. So they can apply for this seed certification process. Okay. So number one, so you have your application for seed certification. So the applicant is required to supply the following informations. So history and origin of the cultivar. Then you have documentation of evaluation conducted in comparison with other cultivars. And then a detailed description of how the class of seed will be maintained. And then you have here the number of generation that can be grown from the breeder seeds. And then the source of seed for planting. And then the cropping history of the land. Okay. So the second step of the seed certification process is the source of seed. So the grower must use a seed class such as foundation or registered seed as an approved cultivar for initiating the seed certification process. And then for the site or the land, so the land should not be a source of contamination from volunteer seeds or volunteer plants or noxious weed. And also the field must not have been grown the crop in the recent 5 years or past unless the previous crop was of the same cultivar and properly certified class. Okay. So the land must not have these noxious weeds and these volunteer crops or volunteer plants. And also, the field must not have been grown of any crop for at least 5 years. Or kung gitamnan man siya, dapat ang gitanom is the same cultivar and the same seed class nga gitanom sa ipa-register nga seeds karon. The next, you have here the management in the field. So the field must be kept free from weeds. Any off types must be rogue out before they flower. Why? Kaya kung i-rogue out nimo siya after they flower, naanay possibility nga naka cross-pollinate na ang mga pollen aning rogue or kining unwanted or undesirable crops or undesirable plants base naka-pollinate na ni sila. Mo nang dapat i-rogue out sila before or tanggalon sila before they can flower. So here, the fifth process, you have here the field inspection. So the inspector, an authorized personnel from the seed certifying agency, looks for the presence of noxious weeds in the field, seed-borne disease, and any factor that might affect the purity of the cultivar. So upon growing of the seed in the field, na ay mo visit nga mga inspector. So they will look for the noxious weeds, seed-borne diseases, and any factors or diseases that can affect the purity of the cultivar. The next, harvesting and processing. So the equipment for the harvesting must be thoroughly clean to avoid mechanical mixture from occurring. And then the harvested seed is cleaned, conditioned, and bagged. So next, let's proceed to the last step in seed certification process. So you have here seed testing and release of certificate. So the representative samples are drawn from each lot by the official of the seed certifying agency for seed testing, which includes the following. So here, the seed inspector will take samples from each seed lot or each lot, and then these seed sample will be tested for the following. So number one, you have the seed germination test or the test for viability. So seed viability is determined by conducting standard germination tests to determine the germination percentage or the percentage of normal seedling produced by the pure seeds. Then you have here, second, purity test. This can be done with physical and genetic tests. So the sample seed should reflect the physical feature of the cultivar, and for the genetic test, so a genetic purity test required by a chemical test that may be as relative sample of isozyme relatively simple as isozyme analysis or sophisticated DNA profiling or DNA fingerprinting. That is for the genetic purity test. Then next, you have here the vigor test. So seed vigor test determines the capacity of the seed to emerge rapidly and uniformly and develop into normal seedlings under a range of conditions. Then you have here seed health. So seed sample is examined for the presence of pathogen by visual inspection, seed incubation, or biochemical testing. And then last, you have here test for noxious weed seed. So the weed is officially prohibited from being introduced into production region. So that's why it is included in the seed testing. So this can be done through physical examination of the seeds for the presence of these noxious weed seeds. The next, once the seed meets or exceeds the minimum standards set by the BPI, the seed is declared officially certified and receive the official tag indicating the result of the seed analysis, which includes the percentage germination, inert matter, weed seed, if there is any weed seed, and many other information about the seeds. So now let's proceed to the classes of seed from these seed certification classes. So you have here classes of seeds. So you have number one, the breeder seed. This is the seed of a new variety that has the highest purity and is produced and developed, controlled, and provided directly by the breeder or the institution for further multiplication. So the tag for this breeder seed is white. So the one who maintains this breeder seed is the breeder itself, the one who developed the seed, or the institution. So they are the one producing these breeder seeds or multiplying these breeder seeds. The next, you have the foundation seed. So this is the progeny of the breeder seed produced by trained officers of agricultural station in conformity with the regulated national standards and handled to maintain genetic purity and identity of the variety, and it has the red tag. So foundation seed, these are maintained by trained officers of agricultural station so that these trained officers maintain and conform with the regulation of national regulation standards to handle the genetic purity and identity of the foundation seeds. Okay. The next will be the register seeds. So register seeds, this is the progeny of the foundation seeds grown by selected farmers and handled to maintain genetic purity and identity, and has undergone field and seed inspection to ensure the conformity with the standards. So it has the green tag. So in here, from the breeder seed that is maintained by the breeder itself, foundation seed maintained by trained officers of agricultural institutions. And then for the register seeds, you can have here grown by selected farmers. Okay. And then last, you have here certified seeds. So this is the progeny of foundation or either registered or certified seeds handled to maintain sufficient varietal identity. So when you say sufficient varietal identity, it must have this uniform or the same maturity, the same plant height, the same heading dates. So they have this, they still maintain the sufficient identity of the variety and the purity grown by selected farmer under prescribed conditions of culture and isolation and subjected to field and seed inspection prior to approval of certifying agency. Then harvested from this class is used for commercial planting. And then you have here the blue tag for the certified seeds. Then you have here the references that I have used in this particular PowerPoint presentation.